Welcome to the Biology 341 Evolution and Population Genetics lecture on genetic variation and mutation. So today we're going to introduce the concept of mutation, why it's important for evolution, talk about the types of mutations, we're going to do a little bit of mathematical modeling of mutations in a population, and then talk about mutation rates. Now, genetic variation is the substrate of all of evolution. If you don't have variation, then you can't have natural selection or genetic drift or gene flow. You first need to have something that is creating genetic variation, and that is typically mutation. There really are two major sources of genetic variation and novelty in evolution. The first is mutations. Mutations are really the only true source of novelty and variation in evolution. However, we can also think of sex as providing novel genotypes by through genetic recombination, basically shuffling all the different combinations of alleles that you can have along a chromosome. Sex does bring us novel genotypes and allows us to explore genotype space, but mutations are the only things that create novel alleles in the first place. So sex, just to reiterate, sex creates genetic variation by giving us different combinations of little a and big B by putting, and big C. So by putting little a and little b with a large C, we create a brand new variation. However, today we're going to be talking about simply new mutations rather than recombination. So today we'll be talking about the very basic type of mutation as well. These are point mutations. We can think of these as either single nucleotide substitutions, so changes just from an A to a G, or from some base pair to a different base pair. Or we can think about insertions and deletions, in which we insert or delete a certain number of base pairs. There are also mutations that can happen at the genome level, so at the gene level, the chromosome level, or the genome level. We'll talk about those in the genome evolution lecture. So, types of mutations. We can broadly break down mutations into two major groups. And there's a lot of different ways we can break down mutations, but this is one of them. And we can think about mutations that happen that are structural, that actually happen in the coding region of the gene, where they actually change the amino acid composition of a protein. These changes can then alter the secondary, tertiary, or quaternary structure of the protein and alter its function. The other category of mutations are regulatory. These are changes in how the gene is expressed. So if you have a mutation in a promoter region, you might enhance or repress the amount of expression that gene undergoes. So just to recall, mutations that happen within this open reading frame of the actual gene, those would be structural mutations. Mutations that happen in the regulatory sequence, in the enhancers or the silencers or the promoters, those are going to be regulatory mutations. Another example of a regulatory mutation would be lactase persistence. So lactase is an enzyme that is present in human babies which breaks down lactose in mother's milk. Once it breaks down lactose down into component parts, galactose and glucose, these smaller sugars can then be absorbed. In the ancestral human population, lactase expression is turned off as the child matures. So an ancestral human who drinks milk is not able, an ancestral human adult who drinks milk is not able to break down lactose and will experience the unpleasant side effects of lactose intolerance. Now, for some populations of humans that have domesticated cows, and this includes Europeans as well as a number of different African populations, a mechanism has turned off this lactose intolerance. It's basically allowed the expression of lactase in adults and therefore they're allowing adults to process milk and process the sugars within milk. And for populations that domesticated cows, there would naturally be positive selection for adults that could drink and get energy from the cow's milk. So when the ability to process lactose with lactase persists into the adult stage, we call this phenomenon lactase persistence. Sarah Tishkoff and colleagues have shown there is not just one mutation, but 4 different mutations that are somehow associated with lactase persistence. We've known for a long time that there is a mutation in intron 13 of the MC-M6 gene. This mutation in Europeans is strongly correlated with lactose tolerance. So Europeans who have this mutation are able to produce lactase as adults. What was very cool about Sarah Tishkoff's work is that she showed that there were at least 3 or 4 other mutations that occurred in other regions within the MC-M6 gene. And these mutations occurred in different populations within Africa, and all occurred within the last 3,000 to 7,000 years, so about the time that humans were domesticating cows. And each of these mutations has happened in a regulatory framework in the intron rather than the expressed part of the gene, and alter the expression of lactase, allowing lactase to be expressed in the adult. They have all occurred in parallel, apparently. So each population of humans, Europeans as well as Africans, have developed these mutations separately from each other and have developed lactase persistence separately from each other. An example of a structural mutation is the classic example of sickle cell anemia. So we have a basic change from an A to a T. So this A changes to a T and then takes an A. So the RNA that's built from this sequence is GAG, which translates to glutamine in a normal hemoglobin molecule. In the sickle cell variant, we have GUG, which translates to valine. And in a normal hemoglobin molecule, you have your glutamate right here, and the 2 subunits stick together and form a coherent cell. But in hemoglobin, in sickle cell hemoglobin, we have these valines, which causes the 2 subunits of hemoglobin to not go together quite as well. And in situations with low oxygen, we see the cells become sickle and get stuck in capillaries and further reduce the oxygen content of the blood. We can also subdivide all mutations into germline versus somatic mutations. So we can distinguish between mutations that occur within our body but are not passed on within our germ cells versus mutations that actually occur in our germ cells. So the mutations that matter in an evolutionary sense are those that get passed on to the next generation. So from an evolutionary perspective, we kind of don't care about the mutations that occur within the body, within each of our body. For example, cancer mutations won't necessarily be passed on to the next generation. And the type of mutation, as I said, that we're going to talk about in this lecture is point mutations. And this is just basically an error. So you have your helix unwinding for DNA to replicate itself. And as you have DNA synthesis underway with DNA polymerase coming along and writing new chains of DNA, you end up getting a mismatch, just a mistake that's made by the enzyme where it accidentally sticks in an adenine to match with a cytosine. Now these two don't go together, and that's how we know it's a mutation. An example of something that could cause a point mutation would be an alkylation event. Here we have, in the presence of an alkylating agent, we have guanine with 3 hydrogen bonding locations. We have the alkylating agent coming in and alkylating the top binding site where this oxygen is, and creating a guanine that only has 2 binding sites. So to DNA polymerase, this guanine actually looks like an adenine. And because it looks like an adenine, DNA polymerase will stick on a thymine here instead of a cytosine. Now, this is an example of an alkylating agent, mustard gas. This young man is in World War I was subjected to alkylating mustard gas, and it caused mutations and burns within his body. Now, it's important to note that so long as these mutations did not affect his sperm cells, he will not pass these mutations on to the next generation. So these are somatic mutations. Now, it's important to remember that there are 2 classes of nucleotide. There's adenine and guanine, which have 2 nitrogenous rings and are considered purines. And then there are cytosine and thymine, which each contain 1 nitrogenous ring and are considered pyrimidines. And so adenine base pairs with thymine with 2 hydrogen bonds. Guanine base pairs with cytosine with 3 hydrogen bonds. Now, because adenine and guanine are both purines with 2 nitrogenous rings, it is more likely that a guanine is going to change into an adenine than a guanine is going to change into a cytosine. Similarly, because cytosine and thymine both have a single nitrogenous ring, it is more likely that a thymine will change into a cytosine or vice versa than a thymine would change into an adenine or guanine. We call changes between the 2 purines transitions, and changes between the 2 pyrimidines transitions, and changes between a purine and a pyrimidine is called a transversion, and is much rarer to see in DNA data. Just to remind you of the central dogma of molecular biology, information flows from DNA to mRNA to protein. And when we're transcribing and translating DNA, the tRNAs that are involved in that compose this genetic code. But if you notice, there's actually 6 different ways to create a leucine. So there's UUA, UUG, CUU, CUC, CUA, and CUG. All of these different ways, all of these different codons code for exactly the same product. So this is a huge amount of redundancy. If you change the first base pair, you can actually change it from a C to a U, and it won't affect the leucine product. If you change anything about the second base pair, then you'll see that you're going to create some different product. So if you change the U to a C, then you're going to end up with UCA, and you're going to have a serine product. However, you can make any change you want in the third base pair of the codon, an A, a G, a U, or a C. Any one of those will still produce a leucine. So it's very redundant in the third base pair of the codon. And this redundancy extends to all the rest of the genetic code. So there's actually at least 2 different ways to say every single amino acid, except for tryptophan, which has only 1 possible codon. All the other ones have at least 2 different codons that can code for exactly the same amino acid. There's also 3 different ways to code for the termination of transcription. So there's 3 different stop codons, and there's 1 single start codon, AUG. And so this is, so we have about 64 different ways to talk about basically 21 or 22 different possible outcomes that we want to have in our genetic code. So this is extremely redundant. Some terminology. mutations that lead to amino acid changes, so typically changes in positions 1 and 2, if you have a change from a U to anything other than a C, you will change this leucine to something else. Similarly, if you change anything about position number 2, you'll also change leucine to something else. These changes are called non-synonymous substitutions. Mutations that do not lead to amino acid change, this is usually position 3, these are called synonymous substitutions. So why does this redundancy exist in the genetic code? I encourage you to pause the lecture here and think about it for a little bit. Try to figure out why you would have so much redundancy in the genetic code. The major reason that we have so much redundancy in the genetic code actually is basically mathematical. If we assume that life needs all the 20 amino acids that are commonly seen in nature, so you can't have life unless you have all of those 20 amino acids, then you need to be able to code for all of them in the genetic code. With 4 possible bases, if you have 1 base pair per codon, so if you basically were trying to code for an amino acid with a single letter, there's only 4 different types that you can code for, A, C, T, or G. Each one of those could code for an amino acid, but you'd only have 4 choices. If you have 2 base pairs per codon, so your codon is made up of 2 letters, an A and a T, or a C and a G, that still only gives us 4 squared, or 16 types of amino acids. So it's not enough to cover all 20 amino acids. If we have 3 base pairs per codon, as we actually do in nature, then there are 64 possible combinations. This is enough to cover all 20 combinations, as well as provide lots of redundancy. A more approximate reason for why we have so much redundancy is that you could have a mutation that occurred in the third base pair, and it wouldn't affect the amino acid. So having that extra redundancy protects us a little bit from mutations. So there are 3 different ways that point mutations can affect proteins. The first is that they don't change the protein at all. The mutation that we observe is synonymous and leads to the same amino acid. This would be a neutral mutation. It might change it by changing the protein sequence. So if it changes it to a different protein, a different amino acid, then that will change the protein sequence and it will be a non-synonymous change. Or it might add a stop codon, in which case it would stop transcription entirely at the point of the mutation and would basically lead to a nonsense protein. So just to review, 6 different ways to code for leucine. We can change from a U to a C and still get a synonymous amino acid. But if we change from a U to an A in the first position, then we change from a leucine to an isoleucine. So that would be a non-synonymous change. In the second position, any change that we make, it has to be a U in the second position or it also won't be leucine. If we change the U to a C, then we get proline. And finally, in the third position, we have complete degeneracy, complete redundancy, and changes from any amino acid, from any base pair to any other base pair will result in exactly the same product. So, synonymous mutation would be a change from a valine, GTC, to another codon for valine, GTA, and we have the same exact product. That's a synonymous change. A non-synonymous change would be a change in the first position in this example from a GTC to a TTC, which changes it to phenylalanine. And sickle cell anemia is an example of a change, of a non-synonymous change from glutamic acid to valine, as we've already talked about. It's a missense mutation, a non-synonymous change at amino acid number 6 out of 146 amino acids in beta hemoglobin. Finally, we could get a mutation that changes lysine, in this example, to a stop codon from AAG to TAG, at which point it would stop reading the rest of the reading frame and basically lead us to a very shortened protein. This was what we call a nonsense mutation. So nonsense mutations are the cause of cystic fibrosis. Cystic fibrosis is a genetic disease that leads to a number of troubling symptoms. Most troubling is a buildup of mucus in the lungs and trouble breathing due to that buildup of mucus. And it is caused by nonsense mutations, as well as other types of mutations that occur to the cystic fibrosis transmembrane conductance regulator. This is a protein that transports chloride ions out of the cells, and these chloride ions help to remove mucus outside of the cell. When these chloride ions are not transferred out by the CFTR channel, we end up with a buildup of mucus along the cell walls and a buildup of mucus in the lungs of children who have cystic fibrosis is what leads to the most concerning symptoms. And there's a whole variety of different mutations that can occur. So this X means this is a change from glycine to a stop codon, tryptophan to a stop codon, arginine to a stop codon. You can also have a deletion or an insertion, which we'll talk about, or a change from a glycine to an asparagine. So given the genetic code, which codon position do you expect you would find most non-synonymous changes? Pause the lecture here to think about this. I would argue that in the second position, any change that occurs in any second position for any of these amino acids, if you take a look, changes in the second position will always lead to a non-synonymous change. In the first position, you also see a fair lack of redundancy, so that many changes in the first position will also lead to non-synonymous changes. While changes in the third position, there are some changes that can cause amino acid changes, but a lot of the changes in the third position are synonymous. So most point mutations have no effect, and that's because there's about 3.1 billion nucleotides in the human genome, and most of these nucleotides are not coding, and they are not regulatory either. So any mutation that happens in these non-coding base pairs is not going to change the function of the genome. So any mutation that occurs in many parts of our genome is going to be neutral. But it's mutations that affect function that are what matter and are what are important for evolution. So now we're going to move on towards insertions and deletions, indel mutations. And indel mutations typically lead to frameshift mutations. So if we have a typical DNA sequence that is coding for a set of amino acids like this, if we stick in a T, it's going to mess up the entire reading frame and it's going to throw it out of whack. So if we insert this T, we go from CTA, TTA to TGG instead of GGA. So we change the amino acid, and then the next codon downstream is also pushed off of its reading frame. So it goes from a GTT to an AGT, completely different, and we have another amino acid change. And all of the codons downstream of this insertion get their reading frame changed and completely change the amino acid. So a whole bunch of non-synonymous changes until we eventually get to a stop codon, which leads to a nonsense change and terminates the transcription of the product. So a frameshift mutation can really screw things up. Similarly, if we have a deletion, then we move everything out of reading frame in the other direction. And so we end up with GAG instead of GGA, and TTT instead of GTT, etc., until we get to a stop codon. Most frameshift mutations will eventually lead to a nonsense change. So thinking about a metaphor for mutations, if codons are words, codons code for amino acids, and those are words, and the letters are DNA, then a synonymous mutation would be a change from Mary had a little lamb to Mary owned a little lamb. So the same codon, but with pretty much the same meaning. A different codon, but with pretty much the same meaning. a missense mutation. So we add we change had to mad. So Mary mad a little lamb. So that obviously is that sentence does not work as well as the original sentence. And then a frameshift mutation would be like saying Mary, and then you add an M in here, and then all the other letters have to fit into the same word reading frame. So you add a MMA, and then a D, and then a little, and an ilam. So you're basically making a complete nonsense out of your amino acid chain by using an insertion or a deletion. So one way that we can get insertion or deletion is through intercalation. There are molecules that will stick to DNA, and they will be read as an inserted base pair by the DNA polymerase. Great example of this is Ethidium Bromide, which is a chemical that is used to label DNA in molecular labs. It fluoresces under UV light, and it intercalates itself into DNA, so it allows us to detect when DNA in large amounts is present. However, because it's also in an intercalator, it will actually, if we expose ourselves to it, could potentially add an extra base pair, an insertion, into our DNA, thereby leading to nonsense mutations downstream. Another type of damage that can lead to a frameshift mutation is UV damage. UV damage can cause thymines to dimerize so that they look like a single thymine instead of two thymines. And this will lead to ultimately to the deletion of one of these thymines in the DNA sequence. So again, either an insertion or a deletion of a single nucleotide is going to change the entire reading frame and lead to nonsense mutations. If we add two nucleotides into the reading frame, a TT, or we delete a GG, that will also completely screw up the reading frame and generally lead to termination codons and nonsense proteins downstream. However, if we add 3 base pairs in, then we're simply adding another codon. The rest of the codons downstream are not affected because the reading frame hasn't been thrown out of whack. You add in an extra amino acid, but you still have the same product downstream that you originally had. It's just bumped by one base pair, by one amino acid. So an example of a frameshift mutation is Tay-Sachs disease. Tay-Sachs disease basically causes a progressive deterioration of nerve cells and mental and physical abilities that begins around 7 months of age and results in death of the child, usually by the age of 4. And it results from an insertion of 4 base pairs within the hexa gene. So we have an insertion of TATC, which throws the entire amino acid chain out of whack until it eventually reaches a stop codon and a nonsense protein is created. So mutation is a double-edged sword. Most mutations are neutral. Most mutations have no effect on fitness. They don't change proteins. They don't change regulation of proteins. They just occur in a part of the genome that's non-functional. But non-synonymous changes that affect functional genes, most of those are going to be harmful. Mildly deleterious mutations might hang out in a population if selection isn't very strong against them. They may be able to stay in a population for a little bit longer. And recessive mutations will also be able to stay longer in a population, only seen by selection in the homozygote for the recessive allele. When they're heterozygotes, they're masked from selection. And this is just some data about mutations within viruses. And you can see that most of the mutations that occur are neutral. They have no effect on fitness. The next largest category are mutations that are lethal. They change the function of the gene that they're in so much that they lead to a dead organism. There's a number of deleterious mutations that occur, and then also a very small number of the mutations might be beneficial in the environment that the organism is living. So we can test the effect of mutations by using a mutation accumulation experiment. What we do here is we take a single cell from a single colony on an agarose plate of bacteria, and we start a new colony with that single bacteria. And we allow that colony to grow, and then we take a single bacteria from that colony and allow it to and grow it out in culture. Then we take a single bacteria from that colony and grow it out. And in doing so, we basically are maximizing genetic drift. We're not allowing selection to occur because we're randomly picking an individual to reproduce. And we're therefore, because natural selection is not working, we're allowing mutations to build up in the lineage of these bacteria. So we're not allowing the mutations to be removed by natural selection, and eventually we can get, for example, 3 or 4 mutations in the population, and it basically causes these populations to accumulate mutations over and over again. And if we look at the fitness of a mutation accumulation line versus a control line that did not have mutation accumulation, we see that when you accumulate mutations, they typically have a deleterious effect. They will result in less survival to maturity over time. However, the very small number of mutations that are favorable for a particular environment are the entire substrate for adaptive evolution. It is selection on these favorable mutations that leads to adaptation. So mutation is a relatively weak force of evolution when acting by itself. We can create a mathematical model of how mutation affects a two-allele system, the same type of two-allele system that we were using for Hardy-Weinberg. So in this two-allele system, we have two alleles, A1 and A2. A1 mutates to A2 at a rate of P, where P is the frequency of allele A1, times mu. And A2 mutates to A1 at a rate of Q, which is the frequency of A2, times nu. And then after one generation, P prime, so the value of P, the frequency of A1 allele, after one generation is simply P prime times P times 1 minus mu plus Q times nu. So we're having an increase. This is how much P is increasing. This is how much P is decreasing. And it's relatively simple to pick a realistic but on the fast side mutation rate. So if we pick a fairly fast mutation rate of 1 times 10 to the negative 5th mutations per generation, and we have that as our mu and our nu as twice that, 2 times 10 to the negative 5th, we can stick that into the equation and see that mutation over a single generation changes the frequencies of alleles in populations almost not at all. And we can take a look at how this works in a simulator. If we look at allele A1, this will simulate mutation over time. So if we stick in a mutation rate for mu of 10 to the negative 5th, and for nu, we let this run for 1,000 generations, you can see that there's very little change in allele frequencies over that time. We get about 0.5 percentage change in the frequency of A1, which increases over A2 by about 0.5% after 1,000 generations. So mutation by itself is a fairly weak force of evolution. We can also figure out what the equilibrium value of P will be if we both know the rate of mutation from A1 to A2, which is mu, and from A2 to A1, which is nu. It's simply nu over mu plus nu. So if mutation from A1 to A2 occurs at twice the rate of mutation from A2 to A1, then it will converge at an equilibrium value of 0.33. Okay, let's think a little bit more about mutation rates. Mutation rates are very different among species. As you get sort of higher in the tree of life, you find that more complex organisms have DNA polymerase that has better proofreading capability. And so you see the mutation rate generally decreasing as you get more complex. In HIV, we have a very high rate of mutation. It's basically the rate that I used in our previous calculation. So 1 out of 10 to the negative fifth base pairs per generation, that yields about 1 error per genome per replication cycle, because they have about a 10,000 base pair genome. They replicate themselves very much over the course of a single day. About 1 billion cells can be infected in a single day, and because there's so many HIV virions and such a high mutation rate, every possible point mutation will occur in an AIDS patient, and every possible one will occur 10,000 to 100,000 times per day. So viruses are extremely adaptable because they're able to experience every possible mutation thousands of times a day. It's much lower in most other species, including bacteria, which have 1 error in every million or so base pairs per cycle. Fruit flies, 1 out of every few hundred billion. That yields about 120 new mutations per genome per generation. So if we look at the rate of mutations, where we have genome size on the x-axis, genome size in base pairs, and we have per base pair replication on the y-axis, you see RNA viruses have very high mutation rates, then other DNA viruses have lower mutation rates. Bacteria and archaea have even lower mutation rates. Eukaryotic microbes have even lower, and then we get all the way down to humans, which have among the lowest mutation rates on a per base pair basis. However, humans and other eukaryotes have very large genomes, and because we have such large genomes, we actually experience more mutations per generation. So each of us is born with up to 100 or more mutations in our genome as relative to our parents. However, we can sustain this high mutation rate because we have a lot of junk DNA, a lot of DNA that isn't coding, and so those mutations are generally neutral. And mutation rates can evolve. So high mutation rates can be advantageous for things like viruses that are constantly in stressful environments that are adapting to them even as they are trying to adapt to the environment. In the face of environmental stress, bacteria will develop strains called mutator strains, which have an elevated mutation rate. And selection might favor less accurate DNA replication to elevate the mutation rate. It's also worth pointing out that because our mitochondrial genomes are prokaryotic in origin, they have worse error-proofing mechanisms, and so our organelle genomes, such as mitochondria and chloroplasts, have higher mutation rates than the nuclear genome. And then there are some parts of the genome that also can have elevated mutation rates. So remember that mutations in the germline get passed on to the next generation, and you can have about 100 new mutations per individual. And out of those, about 1.6 of these mutations will be deleterious mutations that occur in protein coding sequences. Other more harmful dominant mutations will immediately lead to the death of the organism that they occur in, and therefore they will be selected out of the population. Whereas recessive mutations can stick around longer because they're masked from selection by being in heterozygote form. So stop and think about this for a moment. Would you expect to see sex differences in the mutation rate or in the germline, and why? Pause the lecture now to think about this. So in general, we would expect to see males having higher rates of mutation than females, and therefore males much more likely to develop and pass on genetic diseases, especially with increasing age. Females only make one set of eggs. Those eggs are basically put on pause while the female develops, and there's never another set of gametes that are made in the female's lifetime. For males, sperm production is ongoing, and every time there's a cell division, there's an opportunity for a mutation to occur. So males are much more likely to have this replication error that leads to mutations. So mutation rate in females is constant with age because all of their eggs are created when they are embryos. For males, the mutation rate increases exponentially with age. So we see a much higher mutation rate in males that are 45 versus males that are 30. So it could be argued that men are introduced the highest levels of mutations into the population, and especially older males are sort of a health hazard to the human population in terms of bringing in genetic mutations that might be deleterious. There's a wide list of mutations that have an association with paternal age. So the older the father is, the more likely infants are to show these various diseases. You can see an increase in autism, schizophrenia, and other diseases that has a clear trend with older and older males. So if males are experiencing a higher mutation rate in the germline and are also undergoing most of the selection, and we'll talk about sexual selection in a future lecture, then it's possible to conceive of the idea that males are driving much of the evolution that we see in human populations.